Okay, pupe folks, I was just working on this project and I thought I'd make a quick video here while these ideas are fresh in my head and kind of pass them on to you guys, and then we can talk about it in lab meeting or something. So this is something we can do for our poster. I know we didn't really talk about this in the abstract, but that's okay. It's okay if you change your poster a little bit from the abstract. People usually don't get too mad. So this is, you guys gave me the idea for this. Remember the article you showed me during lab meeting where there was a graph of the pupil position, or it might not have been lab meeting. It might have been just you and me, Abby. I forget when this was, but somebody showed me a article where they were looking at the pupil size and there was a graph of it, and it gets smaller when the stimulus is shown, and then it kind of gets back to normal. And this was slower to occur in depressed people or something. The back to normal thing was slower. So I thought that would be a great thing for us to do. I hated what we were doing before. I mean, I didn't hate it, but I realized it was limited because we have so much data, so many data. We have thousands of data points, and we were essentially averaging, a few hundred at the beginning and a few hundred at the end and saying, that's the truth. This one number that is the percentage difference between the beginning and the end. And our data are so rich. We have the pupil, an actual pupil size at every frame for like 10 seconds. And so why not look at that rich data set altogether? And I had wanted to do something like that, but just seeing it in that other paper made it clear. Why don't we just do this right now? So I made MATLAB generate some files for us from our data, like we had been doing before. And what those files look like is over here. See, it says end pupe mat 474. So this is for subject 474. And when I load this in, like I just did, it creates one variable, end pupe mat. So it's a matrix. Mat means matrix, and it's the last little bit of pupil size for a given trial. So now, so I can do maybe both. End pupe mat, well, no, let's just start with that. So I've got this little script here. We can have end mean and end. I think I meant to grab one of the starts to show you guys, but I actually only grabbed an end, so we'll just do an end. Okay. All right, so I'll comment this out, and then we'll run our program. Oh, sorry. So when I type in end pupe mat, look what it is. It's this big matrix. Oh my gosh, 240 columns. Huh. A lot of them are nans, but a lot of them are numbers. What is this? What is this huge matrix? See, columns 116 through 138 are here. Each column has 40 rows in it. So actually what this is the last 240 frames of data, the last 2 seconds of data, because there's 120 per second, for each trial. So each trial, so this is one trial here. And then this is, the fourth to last trial here. And we have each one shown. So what I did is I just, in this little script here, took the average of those, the average across all trials for one subject, and the standard deviation. And then what we can do is make a graph. Oh, you make me so sad. Okay, we have a T in there. Where's the T? Right there. Get out of here. And look at that. That's the average for this one human being. The average of what their pupil is doing at the end of the trial, the last 2 seconds. Oh, that's so pretty. And we can see the standard error. We can see how much variation there is. So you can see it gets a little wonky at the end here. The standard error is growing. But it's still fairly reliable. I think it's not totally off the charts. So essentially the pupil is constricting a very small amount, maybe 3% or something. It's going from 38 to 37, maybe 2.5%. Okay. Cool. Let's see what it does at the beginning of the trial for this person. This is, what we just looked at, 474, right? Let's look at start pupevid. Start pupevid, okay. I'll uncomment that. In fact, let's do it like this. We'll say figure. Sorry, my tea's boiling. We'll say figure, and then we'll do a little plot where we'll have, actually, yeah, we'll just draw them both on the same plot. Oh no, that's not going to work. Is it? Yeah, not going to quite work. So we'll do two separate plots. So we'll do a subplot. So this is telling MATLAB, I want you to make, within this figure, I want you to make subplots. 2 rows of them, or let's do 1 row, 2 columns, and this is the first one of those. So that's what the syntax means. It means 1 row, 2 columns, this is the first one. And then I'll say subplot 1, 2, and this is the second one of those. And if I hit play, it should just nicely draw. Okay, look at that. Oh my goodness. This is so pretty. So this is what it does across all 40 trials for this person at the beginning. There's this big waka waka drop. And then it kind of grows to be about 37. For this person at the end, it kind of goes. Maybe we should make these scales the same, I think. Definitely. Oh, yeah. Let's make the scales the same. So we'll add this axis thing. Go 0 to start mat, I think, how many? It's 5 seconds or something, so 0 to 650. And then we'll go from 0 to 75. I don't know. Let's try that. Look at that. Oh, that's so cool. So basically it doesn't do too much. It does kind of shrink a little bit at the end, but This is really cool. So this is just one person. But averaging across 40 trials and with it's not like a 0 or a 1. Each trial we have a pupil size, which is a dimensional variable. So it's very powerful, very rich, this data set. So I wonder if we can kind of then take the average of all the people who took oxytocin and all the people who didn't, and see if this little curve here at the beginning is different for oxytocin versus not, or depression versus not. Or we could quantify this dip and see how much this dip is and see if that's different in people with oxytocin. So some things to explore. We'll have a lot of fun. And we can put this in our poster, maybe a nice little average graph or something. That is all for now.